Hello, my name is Elias Awikeh, and today I will be presenting about the modeling of glycolysis oscillation within dynamical systems. To begin, I'm going to discuss about what a dynamical system is, because it's a bit of an odd name and it's not exactly very recognizable right off the bat. So essentially, what a dynamical system is a system in nature or a being in nature that shows dynamic properties and that follows laws or rules that go along with it. So that'll make more sense as I explain and go through. There's an emphasis on understanding the geometrical properties and trajectories of the long-term behavior. So later on, we're going to go over different trajectories and patterns that some systems hold in nature and in mathematics. And with further development into research about chaos, which chaos is a huge portion of this, and other factors, dynamical systems has gained a considerable amount of popularity over the last 50 years because of the, evolution of technology. And the definition of chaos in this case would be random states and just things that we would perceive as having no pattern or no fluidity. And as you can see, this is an example of a chaotic system, and the trajectories are what create those two loops, and that small purple ball is just outlining the different loops that it goes through, and we will go over what trajectories and what the small ball is doing later on. So examples of dynamical systems in nature, because we mentioned how dynamical systems in mathematics apply to nature a lot in the last slide, are celestial bodies and waves. So by celestial bodies and waves, I mean planets orbiting around a sun in a planetary system, and waves as in the motion of water in the ocean. So when we look at these sort of things, we have to analyze if there's like a regular or predictable behavior being exhibited, because that will find an easy pattern to analyze, but there's not always a predictable pattern to find. That's part of why dynamical systems is a thing. Sometimes the movement seems completely random, and we need to analyze the random movement to try to find a pattern. And sometimes it's going to be very simple, and sometimes it's going to be extremely complex and need more complex mathematical equations to be able to plot it. And as I just mentioned, it's found within mathematical functions, and typically it uses fundamentals from differential equations and linear algebra, such as matrix algebra and integration and all these different functions. So as I mentioned prior, this is a diagram showing a wave and how the randomness of a wave can be plotted. And just a bit of a background, this is an extremely complex example, because you see there are many different circles, and the dots are circling around the circles, and we'll discuss what those are. Those are essentially limit cycles, and they're rotating around following the motion of the ocean, and it's what perceived to be extremely complex can now be solved into a pattern. And as I previously mentioned, here are the celestial bodies rotating around the sun. And as you can see, the colored lines are their trajectories. And trajectories plays into the geometrical aspect of all of this. And it helps to outline the pattern that was found mathematically. Before we get into all the different concepts, I'd like to just lay them out in a sort of background as basically what we'll be going over. So the multiple topics we'll be covering are linear systems, nonlinear systems, fixed points, stability, and limit cycles. Now, all these are different topics within dynamical systems that we've learned in the class, but they all come together to show how glycolysis works into dynamical systems and how glycolysis has been plotted mathematically to find the pattern. So to start, we're going to go over linear systems. Essentially, a linear system and dynamical system is a dynamical system whose evaluating functions are linear. And an example of that is x dot equals ax plus b, by, and y dot equals cx plus dy. So we don't have any exponents greater than 1 or any random variables or random functions such as e to the x. So this is considered linear because the forcing functions are linear. Linear dynamical systems are solved using the eigenvalue method. So nonlinear systems and linear systems are solved differently, but in this case we're going to be using the eigenvalue method. And for those who do not know what an eigenvalue method is, it's an eigenvector and an eigenvalue is a non-zero vector that is mapped in a given linear transformation. So this is a linear algebra slash differential equation method, and it essentially helps us to map out the vectors and better able to find the patterns within a system. Now we'll get a little bit into fixed points and how those work. So a fixed point is an element of the function that is mapped to itself by the function. And a fixed point within the system can hold stability or instability. So essentially, in a 2D system, as shown in the figure to the right, fixed points can be when the functions intersect or when the functions are at 0. It all depends on the specific system. But the important part about this is the stability and instability. The stability and instability changes the way that the trajectories act within the plane. Stability, in this case, is a condition in which a slight disturbance in a system does not produce too disturbing an effect. Now, if that doesn't make too much sense, mathematically, the function at the point is either moving closer or further away from the origin. And if that doesn't make enough sense, here's sort of a diagram. So look at this lovely couple at the bottom of the PowerPoint slide. Let's equate their relationship status to stable or unstable. If their relationship is unstable, she'll probably be running away and not want to be with him, right? Moving away from him. But if they are in a stable relationship, she is moving towards him, wants to be with him, hopefully. And so it's the same thing with a fixed point instability, or a function instability. Basically, if it is stable, it will be moving closer to the origin, while if it is unstable, it is moving farther away. Now that we understand how linear system works and fixed points work, it's time to go into nonlinear systems. So nonlinear systems are essentially a more complex version of the linear systems. They have functions within them or exponents that a linear system would not have, such as maybe x squared or e to the negative y or e to the negative x. So what would make a function nonlinear is the same thing that would make a nonlinear dynamical system as well. So analyzing these systems within nature can be complicated. Therefore, we need more than just a linear system. So essentially, linear systems can only encapsulate so much of what happens in nature. So to substitute for that, we have nonlinear systems which expand on linear systems and try to cover more of what happens in nature than just a linear system can do. So the method for solving these systems are first finding the fixed points, then finding the Jacobian, and the null lines. So as previously mentioned in the last slide, we discussed nonlinear systems and the tools we need for them. So now we're going to go over the tools briefly because we will be using them later on to solve the nonlinear system of glycolysis. So let's just begin with going over what a Jacobian is. So it's more like a Jacobian method. It's where you create a vector made up of first order partial derivatives of the two functions that you're working with. So for example, if you have x dot and y dot, it would be made up of, as you can see, the top left would be the derivative of x from x, the top right would be derivative of x by y, the bottom left would be the derivative of y by x, and the bottom right would be the derivative of y by y. Now we do this to sort of in this case, we do this to identify the fixed points as stable, unstable, or saddle points, which is very helpful when creating closed systems and modeling trajectories within the model. Next are the null clines. So null clines are essentially plots found based on x equals 0 and y equals 0, and these functions act as a shaping region for the trajectories, as you're going to see. So here's an example down here. So with the plot comes two null clines. In this case, it's x equals 2 and y equals 0. The null clines sort of shape where the trajectories will be, because the trajectories will most likely be in 1, 2, 3, and 4, moving around those areas. It is important to note that trajectories can move between the null clines, but what null clines are mostly used for is a tool in order to plot trajectories, because trajectories can get quite difficult to plot blindly. So now we're going to review a little bit of limit cycles. So limit cycles analyze nonlinear dynamical systems in polar coordinates. So if you don't know, polar coordinates are coordinates in terms of r, radius, and theta, the angle. Rather than having null clines on a plot, there are limit cycles that close the trajectories in, and I'll show you that in a moment. So as we spoke about previously, null clines kind of shape the graph a little bit, but in this case, limit cycles close in on the graph on the fixed points in a rotational area. The trajectories will either spiral towards or away from the limit cycle, and that helps to determine the stability. There are three examples of trajectory combinations: stable limit cycle, unstable limit cycle, and half-stable limit cycles. As we can see, we have three graphs right here below, and as they draw in, the first one is stable because, as you can see, the trajectories are going into the limit cycle. And in the second graph, we have both of the trajectories going outside of the circle, away from the circle, so that makes it unstable. And the third is a half-stable or saddle node. We have one going in, one going out. If it were the other way around, it would still be the same thing. It applies for both, but these are the three different kinds of limit cycles that you will see. In case it was confusing prior, this is what an unstable and a stable limit cycle looks like. So you can see in the first diagram, everything is going towards the circle, making it stable, and in the second diagram, everything's moving away from the circle, making it unstable. So now that we've gone over all these different methods and a background of dynamical systems, it's important to discuss what is glycolysis. I mean, we haven't discussed that yet. So glycolysis is a series of reactions that extract energy from glucose by splitting into 2,3 carbon molecules called pyruvates. So that obviously did not make a lot of sense for us. So it's essentially living cells obtaining energy. So it can be broken down into two phases where it takes in energy and creates energy. These two energies are not the same. It's essentially like eating food and that food being turned into energy. So this is how the cells in your body create the energy that you use, the way that you move. You wouldn't be able to do this without glycolysis. It turns the sugar you eat into energy. Now the word glycolysis actually comes from the words glucose breakdown. So the sugar that you eat to create energy is actually called glucose. So when you take in glucose, as we mentioned multiple times already, it breaks down that glucose. And here is a quick diagram in case it was a little confusing. The glucose sugars come in, glycolysis turns them into energy, and they go out and used in the cells of the body. Now, you may ask, why is glycolysis important for us? Like, what does it mean to us? So, we as humans, we are heterotrophs. So a heterotroph is an organism that requires an external supply of energy in the form of food, as it cannot synthesize its own. It needs to eat other organisms, and in this case organisms are like any food we eat, to produce energy, and then once we eat that food, the glycolysis creates the energy. And different heterotrophs in nature are us humans, animals, and many different kinds of bacteria. But you may be asking, what's the opposite? That is an autotroph. An autotroph is an organism that's capable of making nutritive organic molecules from inorganic materials. So if, as an example, a plant doing photosynthesis, as can be seen in the little GIF, a plant eats by taking in sunlight. It's a chemical or light energy reaction. It's not something where it eats something and performs glycolysis. It's more of a completely different process. In this case, it's photosynthesis. Now, what is glycolysis in the sense of what are the steps? What is the breakdown? Now, I won't get too far into it because we are not biology majors. It's not important to us. But essentially, glycolysis is 4 steps: the actual glycolysis, pyruvate processing, citric acid cycle, and electronic transport of chemiotosis. So what we're going to be focusing on for this case is the first sort of step of glycolysis. That is what is modeled in the oscillation of glycolysis. And 2 things to really keep an eye on are ADP and F6P, which stands for adenosine diphosphate, and fructose 6-phosphate. So now that we have all the background that we need, we have the dynamical systems background, and we have the glycolysis knowledge that we just learned, now we can begin to break down what is the oscillation of glycolysis. So the model that we use is the Selkov model of oscillation. So Evgeny Selkov is a computational biologist and mathematician who discovered a pattern of oscillation within glycolysis. Glycolysis was found to act in an oscillatory fashion within intact yeast cells as well in yeast or muscle extracts. So essentially, when the body takes in yeast and eats yeast, they noticed a pattern of glycolysis within the body, and they were able to model that pattern and find oscillation. So the discovery of a waxing and a waning period is actually what caused this sort of oscillation discovery. And without this like sort of switch, Selkov wouldn't have been able to find it. So for the model we're going to be working with, it's going to have X labeled as ADP, adenosine diphosphate, as I mentioned earlier, and Y labeled as F6P, fructose 6-phosphate. And based on all the parameters I just described, the oscillation can be modeled using 2 equations. X dot equals negative X plus AY plus X squared Y, and Y dot equals B minus AY minus X squared Y. So just as a fair warning, we're going to write through this just to have it a little bit easier to explain through writing, and also we're going to try to avoid as much math as possible in order to make it a little more clear, but we will be mentioning math methods that were mentioned in the previous slides. So before we start, I would like to show you a graph of the oscillation that they found. You can see cells performing glycolysis in the left figure, and in this middle figure, you can see a pattern that developed through the ADP and F6P. This is actually glycolytic oscillation in a yeast extract subjected to a constant injection of substrate. So by injecting the yeast cells with trehalose, they were able to find a pattern of oscillation. Now, before we start, it is also important to go over this theorem. This is called the Poincare-Bendixson theorem. It has 4 rules to it, and it essentially goes like this. R is a closed bounded subset of the plane. X dot equals Fx is a continuously differentiable vector field on an open set containing R. R does not contain any fixed points, and there's actually a loophole to that we will be discussing. And 4, there exists a trajectory C that is confined in R, in the sense that it starts in R and stays in R for all future time. So I sort of drew out what I meant here in the bottom figure. You can see R is a trapping region, and essentially what this theorem is saying is that these trajectories in this theorem that pass this theorem are trapped in sort of a region or trapping region of R, and essentially these trajectories will spin around in perfect circles or spin around until infinity, but they will not escape this trapping region. Now, within this trapping region, or this closed region, it is easier to analyze the trajectories within it, and that's why we sort of create these closed regions. But also, we cannot have fixed points within the region as shown by P, but there is a small loophole to that, and that is actually shown by P as well, by having a small hole, and we will discuss that in a later slide. So now we're going to get into the solution a little bit. We have the model that was described earlier, and the first step that Selkov took was finding the nullclines. Now I'm not going to take you through it, I'm just going to describe what the nullclines he found were. In this case, they're y equals b over a plus x squared and y equals x over a plus x squared. And eventually, he plotted them to find this plot. Now, there's a lot going on in this plot, so I will begin to describe it. So, when you plot two nullclines, it sort of separates out the area, and you have to find the trajectories within the areas that are separated out. And you can see that by all the different little vectors that are drawn in. You have it in all the sort of pockets created by these lines. Now, to tell where the trajectories go, you have to plug values in that region into x dot and y dot, and it'll essentially give you a direction based on where that line would be traveling, right? So that's how you create trajectories within the model, but we need to take it a step further, and we need to sort of begin to create the closed region as we discussed, because we need to analyze the oscillation within this closed region. So in order to create the closed region, as you can see by the rainbow trajectories, all the trajectories need to be pointing inwards in a sense. They need to be closing in this particular area to prove that there is a closed area. So the way that Selkov did this was he began by plugging in a very large x, or assuming a very large x, which gave him these approximate equations of x dot equals x squared y and y dot equals negative x squared y. Now, by dividing them, he found that the slope was -1, which means to him that it was almost parallel to that of the trajectory, or the closed region shown on the left. Next, he wanted to he wanted to analyze it in a more precise way. So what he did was he did x dot minus y dot, or in this case, negative y dot, because that's what you get. And he ended up with the equation b minus x. Now, the conclusion he got from that equation is that if negative y dot is greater than x dot, then x is greater than b, and vice versa. So this tremendously helped him to discover the trajectories actually going inwards and closing the region, not just being parallel as we discovered before it became a little bit more precise. Now, what he did with this information was he drew out the region, but the problem was that it is almost a closed system, but it has a fixed point. And as we discussed, the PD theorem cannot have a fixed point within it, or it is not closed. So what we do is we use the small loophole with something called a repeller point. So the fixed point shown by the intersection of the nullclines, we have to be able to prove that this point is a repeller point, and a repeller point is trajectories moving away from the point, causing a infinitesimally small hole. And in this case, this hole is enlarged just for you to be able to view it. So we need to be able to prove that that point is actually a really small hole, and it's not part of the closed system, therefore justifying the closed system. And the way that we do that is we have to prove the stability of the point. And how we prove the stability of the point is using the Jacobian method that we described earlier when discussing nonlinear systems. So the Jacobian method, applying it here, I'm not really going to go through it, but once you apply it and take the two equations that were modeled, you get the Jacobian shown below. And these are the fixed points that Selkov found, x star equals b and y star equals b over a plus b squared. And he was actually able to model a graph based on the constants within the two equations. And based on that equation, he was able to sort of create a plot where the x-axis was one of the constants and the y-axis was another constant. And in the highlighted region, you can see what we are going to be using in this case. But what he found was that in a certain region, it's a stable limit cycle, and in another region, it's a stable fixed point. So as we described earlier, a stable limit cycle is something that has a sort of boundary where the trajectories are going out towards it on the inside and towards it on the, again towards it on the outside. So both the inside and outside are traveling towards that limit. And so it's kind of opposite from what's actually a stable fixed point, because a stable fixed point is negative and it's moving towards the origin, not away from the origin towards a limit. So what we've discovered here is that the highlighted region is positive while the non-highlighted region is negative, creating two separate things, a stable limit cycle and a stable fixed point. And the highlighted region is actually what we will be using, because in this case, if we want to create a very, very small hole at that fixed point, we want the trajectories to be moving away from it, not towards it. Because if they're moving away from it, it can create a very small hole. So anything in that highlighted region is what we will be using. Now, in Selkov's case, he actually ended up using two specific constants, a equals 0.08 and b equals 0.6, which ended up giving him this sort of trajectory plot. And using these two constants, he was able to actually model what happens during glycolysis and the oscillation that happens during glycolysis. And as you can see, just to point out just to really hammer home my point from earlier, you can see the trajectories coming out of the origin or that fixed point between the null lines, heading towards a sort of limit, and then all those others on the outside heading in towards it. So you end up with a limit cycle or a stable limit cycle, which creates the small hole we needed to prove the PD theorem and prove that it is a closed system. In case the presentation didn't convey this point, oscillation within the medical field and finding these patterns is extremely important, and it may not seem as important when analyzing glycolysis because it's something just that the body does, but when you expand, when you start with finding just glycolysis, but then you expand further and you see, wait, what else can we find within the medical field that can do this kind of thing, you discover amazing things. So these are just some things that I found interesting that's sort of related because of the medical field. Something called the high-frequency chest wall oscillation. So just on the topic of oscillation, there's something called cystic fibrosis, which is a disease in which there's a buildup of mucus in the lungs and the digestive system. So essentially using a particular frequency in oscillation, this vest filled with air decompresses of air and fills with air about 20 times a second and causes a frequency of vibration strong enough to sort of break down the mucus and, you know, let it pass through. And like this, as I was saying, like shows that the medical field can be pushed so much further with the use of oscillation and dynamical systems. And not only that, on a more relatable level to glycolysis, there are different breakdowns of and different reactions of elements that actually also incorporate oscillation. So this is just another example of chlorine dioxide, iodine, malonic acid, where just like glycolysis, they were able to take two different elements and assign them to X and Y and actually plot them. And this plot is similar to that of glycolysis. You can see that there's sort of like a little bit of a casing, and two null lines do come together for a fixed point. It all comes together, and it becomes much more expansive. And the reason I wanted to show you this slide is to show you that oscillation within medicine is far more important than we perceive it to be, because as technology advances, we're going to be able to sort of, I guess you could say, cure different things that we weren't even able to think about before. And on that note, that is the end of my presentation, and thank you for listening.